So, this is DNA Technology and Genomics, Chapter 20 in the AP Biology textbook. Manipulating genes. We have to talk about recombinant DNA. That means DNA that has come from two different sources and they're combined or recombined to make something new. It's utilized in genetic engineering and biotechnology. So we're going to look to the bacteria model system for this. And we look at simple circular DNA. Simple circular DNA can multiply quickly. It will pick up and keep, the bacteria themselves will pick up and keep what is called a plasmid, which is a small foreign circular DNA, if it's helpful. An example of helpful would be antibiotic resistance. And scientists have noticed that bacteria do this, and they decided to take advantage of it, if you will. So a gene that confers antibiotic resistance. And we've worked with that when we did our PGAL and PGLOW labs, looking at the operons. We used ampicillin resistance as a way to select the bacteria that had our gene. So in this diagram, you have the bacteria with the bacterial DNA in red and some plasmids in blue circular plasmids. Notice they are indeed smaller. Now let's talk about restriction enzymes. Restriction enzymes are used by bacteria to cut out foreign DNA. So if some foreign DNA does get in, they do have a way, an arsenal, an immune system, if you will, to get rid of it if they don't like it. And it will always cut at specific sites, and they're called, they're usually palindromes, and a palindrome is something like this, GAATTC. If you look at these other strand, it would read the same but backwards, and that's a palindrome. It's a mirror image. And so let's look at the EcoR1 restriction site. EcoR1 cuts between the A's and GAATTC. And you can cut it so that you can insert a gene of interest that you want to make copies of. And you can put it into this plasmid DNA. And there's something called sticky ends. They're overhangs, so it doesn't cut directly through like a straight line. It's a jagged edge. And this will allow you to control how that DNA is inserted into the plasmid. So here's a diagram to give you a better idea. So this GAATTC is cut with the EcoR1, and you notice that we have an overhang with the A's and the T's, and this allows us to insert another piece of DNA in the gray here, that, for example, that it does have the similar ends so that they will fit together, and at the bottom we have our recombined DNA that's got a little bit of our plasmid DNA and a little bit of our DNA of interest. Well, now we can look at something called a RIFLEP. And RIFLEP stands for Restriction Length Fragment Polymorphism. That's a mouthful. Well, what does that mean? Well, restriction enzymes cut DNA into different length pieces. So they'll cut wherever there's a palindrome, and in between those palindromes are different length segments of DNA. So if you look at this diagram, if you cut parents and their offspring, you'll see that they'll have inherited half of each parent's, not only their genes, but their restriction fragments, which are located in the non-coding DNA. RIFLEPs are actually made of VNTRs, which are within the non-coding DNA, and that stands for a variable number of tandem repeats, and they're nonsense sequences. And so if you look at that, what that means, variable number means that some people will have maybe 10, another person might have 20 of these repeating sequences that don't code for anything. They're tandem, so they're in line like a tandem bicycle. An example of a VNTR would be AATTAG. One person might have, like I said, 10 of these within their fragment, and another person might have 20. You can compare these, the RIFLEPs of individuals, in a DNA fingerprint. No two people are identical except for identical twins. And so this is why the DNA fingerprinting has become so important in court cases, because they're very, very, very specific. And we do this by a process called gel electrophoresis. We will be performing restriction analysis, DNA fingerprint by gel electrophoresis after vacation. And so here's a diagram to show you. You load your samples in the wells, which are at one end of the gel, and then you apply electricity, and it will cause the DNA to separate, and it'll go through this agarose, which is it's a seaweed, like a gelatin, and they will separate based on their size. And so if you look at figure 2, you'll notice that there's a bunch of different pieces, different size pieces. The larger pieces do not move as far during the same amount of time, and they will all move from the negative end to the positive end, because the DNA is negatively charged and it is attracted to the positive end. And then we can look at it with the UV light. If we have you have to add a little something to it so that it's visualized under UV light. You can also stain it. We're going to probably use the UV light because we can see it in the same day. This is a sample DNA fingerprint. I made it kind of easy for you to look at. Mom is in blue, and you can see that she has a number of different fragments. And Dad is the yellow, and he has fragments that are actually different from Mom's. They're different sizes. And if you look at daughter 1, she has half of her fragments come from Dad and half from Mom, as you would expect. Same with son 1. But if you look at daughter 2, there's a funny thing going on. She has half of hers match with Mom, and then the other half don't match with Dad. You be the judge. What do you think happened? And same with son 2. He doesn't match with anybody. What do you think is going on with him? Southern hybridization. When you run your samples on the gel, it's really hard to see them because you get so many bands if you think of all the possible DNA. So you transfer the DNA to a special kind of paper. It's called nitrocellulose. And then you incubate it with a radioactive probe. You can also use a fluorescent probe. And this probe base pairs with a specific sequence that you're looking for. And then you can expose this to X-ray film, and then you can see only where the probe stuck. So then you don't have to look at millions of bands. It's easier to visualize what you're really seeing. So this is a diagram from your book of first you run your gel. They put it under the biology book. We actually would do this, use liquid to pull it onto the paper, and you have a lot of padding, absorbent paper to pull the liquid, wick it through. And then you put it in the bag with the radioactive probe, and then it shows that step 5 that you can see the bands that were radioactive. PCR stands for polymerase chain reaction. This is DNA replication in a test tube. It was the guy who came up with it, his name is Kary Mullis, and he won a Nobel Prize for thinking of this idea. And this diagram here shows that you split your double-stranded DNA, and then you add your primer, and then you make copies. You add by adding the bases, and then you repeat. So here's the steps. First, you separate the strands. To do that, you have to heat it to 90 degrees Celsius to separate the hydrogen bonds. Then you anneal at 50 degrees. This is approximate, and it varies depending on the primer, but this allows the primer to bind specifically. And then you lengthen or add all your bases at 72 degrees Celsius. And you repeat this for 25 to 30 cycles so that you get enough DNA. PCR is super important because if you think about finding an eyelash and they can figure out whose eyelash it was, there's not enough DNA in that eyelash. You actually need to make lots of copies of it so that you can analyze it. So PCR has revolutionized our understanding of the world, truly. DNA sequencing is yet another really cool thing you can do. You can determine the sequence of a short piece of DNA. It's by essentially using a similar process to PCR. So you use what's called dideoxyribonucleotides, and they either have radioactivity or a fluorescent molecule in that 3' hydroxyl position. And so once, when the bases that are added in, and it's done by base pairing, that 3' fluorescent molecule or radioactive molecule that's attached at the hydroxyl blocks the addition of further bases. So the fragments will drop off, and you get various length fragments that are made. So let's look at this. So in the tubes, so here's my 4 tubes and for my DNA sequencing analysis. And in each tube, I put the bases A, C, T, and G, just normal everyday bases. I put Taq polymerase. I also put the same primer. And I put my radioactive base. So in my first tube, I have radioactive A. My second tube, I have radioactive C. The third tube, I have radioactive T. And the fourth tube, I have radioactive G. And of course, I have to have my template DNA that I'm trying to sequence. And then basically it works like PCR. And so let's look at the reactions that are occurring in the A tube, the radioactive A tube. So here is my sequence. X-axis is where the primer is. And then I have my bases. Now, of course, I can't see this. I don't know what those bases are, but let's see if I can figure it out. So the first thing that can happen is I base pair, and I start getting a sequence, and I have T, G, G. So you notice that it's base pairing. And then my radioactive A base pairs with the T in this instance. And so no more bases can be added, and this is the end of that segment. At the same time, another piece is being made, and this one goes a little further because I got a normal A in that first position, but at the next time I needed an A, it was radioactive, so I get a slightly longer piece. And then it occurs again, you notice, and I get a little bit longer, and yet another longer piece. So this is all my possible places where a radioactive A could attach. Meanwhile, I have reactions going on in the other tubes, and they are putting in radioactive C's, radioactive T's, and radioactive G's as appropriate for this sequence. I then take all of these and I run them on a gel. And this is what a sequencing gel looks like. And you read the gel from the bottom up, because the shortest piece will move the furthest. Okay? And the sequence will be the complementary strand, not your original template that you're trying to figure out, but it's the one that base paired with it. And so I use base pair rules, and I can work backwards to figure out my template DNA. Over vacation, you have an example to try out. Hopefully you'll get it correct. Transformation, we've already done in class. You're going to put a recombinant plasmid into your bacteria. And when the bacteria multiply, they make copies of the plasmid DNA too. So this is really handy if you want to make lots of copies of, like for example, in this diagram we have human DNA stuck into our plasmid. So we use restriction enzyme, we cut it, we put in our the DNA we want, and then we put it into our cell. We select with antibiotics so that we don't have a lot of bacteria that didn't pick up our DNA. We don't want to waste our time and our money working with bacteria that don't have our DNA of interest. So only the bacteria that have the plasmid that contain the resistance gene against the antibiotic will grow. And we can also use a marker such as beta-galactosidase. That would be that P-gal plasmid that G-period class worked with. You can use the P-glow as well. And those are called marker genes, and allows you to follow the your plasmid because of the expression of the gene. And this is a picture of beta-gal positive bacteria. All the blue colonies are positive. Making a library. So a library is a collection of all the DNA that would be in a given organism. And so a genomic library contains all of the DNA of a given species. That's a lot of DNA, and that includes coding and non-coding DNA. And so we do have a human genomic library, but that's a lot of DNA, a lot to work with, and it makes it very difficult. It's billions and billions of bases of DNA. So a cDNA, which is C stands for copy, is usually made for purposes of determining what the codes of different proteins are that are important. And so it's usually, and this is made from messenger RNA. And I want you to think about messenger RNA. How are you going to, what is that? Well, messenger RNA is what is being made by a given cell, correct? If it's going to be making proteins. So it's important to that cell. Remember that not all proteins are made by all cells, and not all genes are transcribed and translated in a given cell. So, for example, I worked with a rat liver library. I wanted to know only the proteins that were being made in the liver. That was, so I narrowed it down so that haystack, if you will, was easier to find the needle. And I could isolate that messenger RNA, and I'm just throwing this out there, using, remember those poly A tails? Well, the poly A tails on the messenger RNA will bind with a T's. So I would use something, a LIGO DT column. It's a column that has a bunch of T's, and it will cause my poly A tails to bind. So I can isolate all my messenger RNA from my cells. And then I use that reverse transcriptase. And I know we talked about reverse transcriptase with HIV, the retrovirus. And that enzyme will convert my messenger RNA back to the complementary DNA. And then my coding, only the coding DNA is in my library. So I've narrowed it down to just stuff that's actually being transcribed and translated in those given cells. And then I can probe the library. I can screen it or probe it and looking for specific genes that are important in that given cell. And to probe, I use a radioactive complementary sequence for the one I'm looking for. So here we have the filter paper. So I grow up my bacteria colonies. They all contain all possible cDNAs that are, for example, use the liver library here. And then I take my special nitrocellulose paper and I put it down on the bacteria colonies that are on the agar. And then I do a lift. I lift it up. And then I treat it to open up the cells and denature the DNA, because it can't be, if it's bound, hydrogen bound, the double strands are hydrogen bound together, they're not going to be able to bind with my probe, so I have to denature it, and then cause it to stick to the filter, and then I expose it to my probe, okay, and then I take it and I do what's called an autorad, and I expose it to X-ray film, so wherever my radioactive probe binds to it, I'll be able to see. And then I can go back and align that paper with my plate and know what colonies of bacteria have the gene that I'm interested in. And that's the end of the show.